Alright, here we start with Chapter 10, the first section. And Chapter 10 involves looking at antimicrobial treatment, and this is going to be within the body. So Chapter 9 was looking at how we can control microbial growth outside of the body to minimize the amount of bacteria that might get, or microorganisms that get into our body, and then necessitate the need for antimicrobial treatment. So here are your learning objectives for this chapter. 1 through 5 are going to be specific for Section 1. Alright, this first table looks at the characteristics of an ideal antimicrobial drug. Alright, so you want your drug to be selectively toxic to the microbe, but not to the host cell. So if you were to take a drug, the goal would be for it to get rid of the microorganism that is making you sick versus killing you, because that really wouldn't be a good scenario. Two terms that we looked at in Chapter 9 were microbicidal and microbistatic, and we would prefer to have the drug to be microbicidal, that it would kill the organism rather than just simply stop it. Now, if your immune system was fully functional, that might be all right, because if the drug kept the microbe from replicating and just let it sit there, then our immune system could probably mount the proper attack and get rid of it. But for those individuals who are immunocompromised and their immune system is sufficient or severely deficient in its function, then they really need the drug to be microbicidal and actually kill off the organism completely. Alright, it needs to be soluble. It needs to be able to work effectively when it is diluted in body fluids, such as the lymph, the gut, the blood. It needs to be able to get from the oral cavity, from the intestinal tract, into all areas of the body, which means it's going to have to be diluted in blood to be transported. It needs to remain potent long enough to act, and that it's not broken down or excreted prematurely. This is generally why you take a drug 2 to 4 times a day in terms of an antimicrobial. Right? There are some medications like the Z-Pak where you can take several pills for just 2 or 3 days, and that drug is then in sufficient concentration that it will stay in your body for a total of 10 days, which should be long enough to make sure all of the organisms are killed off by that drug. You want it to avoid leading to the development of antimicrobial resistance, which we'll talk about in Section 3, I believe, of this section, of this chapter. And then, similar to the microbicidal versus microbistatic comments, we want it to complement and assist the activities of the host defenses. So really work in conjunction with our immune system. It needs to remain active in tissue and body fluids, so you can't just take it in the mouth or have an oral drug that gets broken down in the acidity of the stomach before it can get into the intestinal tract where it gets absorbed into the body. It needs to be readily delivered to the site of an infection, and it needs to be reasonably priced. This is a big one because there are a lot of drugs that are extremely expensive, and if a person doesn't have insurance, can make it almost impossible for a person to afford the drug that they're needed, or that is needed. And you don't want it to cause allergies or to make predisposing conditions, pre-existing conditions worse, or predisposing the host to other types of infections. Now, here are just some terms that you will want to know. So making flashcards might be helpful here, or simply printing this slide and folding it in half. So then you have chemotherapeutic drug on the one side and be able to recite from memory that it is any chemical used in the treatment, relief, or prophylaxis of a disease. So it's a chemical, it's therapeutic. Prophylaxis is using a drug to prevent an infection. Probably the biggest example of this would be with dental work, which it doesn't happen as nearly as often now. In terms of if a patient has a heart condition, they may go on prophylaxis antibiotics because when you go to the dentist, they may be scraping your teeth to get rid of the plaque and tartar, and in that process create microabrasions to your gums, which then can allow for the bacteria in your mouth to get into the blood. And then they can get into the blood, replicate, and circulate to the heart, which could cause you problems if you are prone to heart issues such as valve disorders. All right, antimicrobial chemotherapy is going to be specific to controlling infection. Antimicrobials is an all-inclusive term for antimicrobial drugs, regardless of its origin. Antibiotics are a byproduct of some organisms that will inhibit or destroy other microorganisms. And semi-synthetic, synthetic drugs are going to be those that are modified in the laboratory. Narrow spectrum or limited spectrum are drugs that have a very narrow effective range. All right, so they're going to be effective against gram-positive bacteria. They're going to be effective against one genus of bacteria. Broad spectrum are going to just be broad. You take it, it kills off a lot of different organisms. And which one's better for you? Well, probably narrow spectrum would be best, because if you take a broad spectrum antibiotic, it's going to kill off the good guys that live in you. The drug doesn't necessarily care if it's causing an infection or if it's benefiting you. If it's broad spectrum, it's going to kill off whatever is in its range. Therefore, narrow spectrum is going to be the best for you. If you can identify very specifically what organism is causing an infection, you can treat it with as narrow spectrum drug as possible. All right, so some questions to ask and get the answers to before you start drug therapy. You've got the what, how, and who. All right, so what is the organism causing the infection? All right, is it bacteria? Is it fungal? Well, if it's fungal, you're not going to treat it with an antibiotic. But you also may want to know the specific type of bacteria that you're dealing with prior to prescribing a drug, an antibiotic. And then, is the microorganism susceptible or sensitive to the different drugs? So you would also like to know or be reasonably confident that the drug that you're giving a patient is actually going to do what you plan it to do, kill off that invading organism. And then who is the patient you are treating? Right, what is the overall medical condition? Are they fairly healthy? Are they already sick with various diseases? Do they have liver or kidney disease? And if that's the case, you may have to be very careful with what drug you prescribe to them, because your liver and kidneys are going to be filtering the medication. And if they can't filter it properly, then maybe the drug gets left in the body at too high concentrations, or maybe the process of actually filtering it will cause more damage than what's already there. All right, this is the Kirby-Bauer test or disc diffusion test, and this is objective number 4 on your list. And what this shows is whether or not a drug is going to be effective against a particular type of bacterium. And so what this shows is, it's easier to see on this picture, you take a sample of the bacteria and streak it across the surface of a petri dish, right? Usually with a cotton swab, a sterile cotton swab, and you can kind of see the streaks here in the bacteria growth, this white part. And right after you streak out the bacteria and you cover the entire surface, you add these little discs to the petri dish, and you can add multiple discs to one dish, And in each disc is a particular type of antibiotic at a particular concentration. And so here you have gentamicin, here you have ampicillin, chloramphenicol, and the different concentrations. And what happens over time is, one, the bacteria grows, which you see here, but also the drug will diffuse out into the media and cause death if the bacteria is susceptible to it. All right, if it is resistant, if the bacteria is resistant, it's going to grow right up to the edge of that disc. And so you know that this is not a good drug to treat this person who has an infection with this organism. All right, susceptible. Enrofloxacin would be a good drug to treat this because it has a very wide zone of inhibition, making it sensitive. All right, another thing that this figure shows, it's kind of hard to see, you have to look at it in a particular light, is that this right here is actually the delineation of the zone of inhibition for this drug, and then this is the circle for this other disc. But right here in the middle is where you should have growth. All right, if you just make a line there, you should have growth right in here. But what this is giving an example of synergism. All right, these two drugs, when combined, actually act against the organism. So even though this concentration of this drug is going to allow the bacteria to still grow, and this concentration right here of this drug is going to allow it, the lower concentration of both of those drugs together, which you find right in here, is actually inhibitory to that organism. So this shows you how or what you can use to treat an organism with. All right, last slide for this section is, and it kind of starts section 2, but looking at the goals of antimicrobial drugs, all right, to disrupt the cell process or the structures of the bacteria, the fungi, or the protozoa. You want the drugs to be able to inhibit virus replication, if that's what you're looking at. You want the drugs to interfere with the function of enzymes required to synthesize or assemble the macromolecules within that organism, because then you can stop the reproduction of those macromolecules or those microorganisms. You want it to be able to destroy structures already formed in the cells, so you don't have to start at, say, ground zero. And then you also want selectively toxic or select toxicity. And this is objective 5. You want to be able to kill or inhibit the actions or synthesis of molecules in the microorganism, but not the host cell. So with bacteria, that's pretty easy because bacteria are prokaryotic cells, whereas we as humans are eukaryotic. And so there's a lot of differences between those two cell types. When we talk about fungal infections, all right, fungal organisms are eukaryotic organisms. We are eukaryotic organisms. So we have a limited number of drugs that we can treat fungal infections because it's just harder to find a target in another eukaryotic cell that is not going to be problematic for the host cell.